Hi, and welcome to In Search of Insight, Nootropics Depot's monthly podcast. I'm your podcast host, Erica, and sitting next to me is our product specialist at Nootropics Depot, Emil.
Hey, everyone.
Today, Emil and I are joined by our science director at Nootropics Depot, Dr. Abraham. Welcome to the podcast.
Hi, happy to be back. And feel free to call me Ellie. We'll keep it more casual.
Awesome.
But the topic of conversation today is about how you did become a doctor and your Ph.D. research. Your Ph.D. research specifically into botanicals and machine learning, and how machine learning can help us identify botanicals better in the future. This could have implications for the whole supplement industry, and specifically for us, because we have a direct source for that research and expertise, because Ellie is on our team now. And we can talk about how all of this will come together and potentially make the products that you're taking even better. Before we start, of course, we like to take something on the podcast. And this was a bit of a last-minute podcast. We are actually going to be talking a little bit about holy basil, so it would have been fun to take our holy basil supercritical CO2 extract. That's an extract that Eric and I are huge fans of, but unfortunately we did run out of it a few weeks ago. We do use it kind of regularly. It's really nice. I'm not sure, Ellie, if you've tried that one.
I haven't tried ours, but I'm a huge fan of basil, and we'll talk all about that. So great. I'm excited to hear you guys actually really like it as much as I would hope.
It's a really nice one.
Yeah.
But for a compromise, which isn't really a compromise, but we're just going to have a nice social beverage, which could be cocoa, coffee, or matcha. We've decided to go for a little matcha drink. It's evening for us. Our energy levels are a little bit lower now, so we need a little bit of a boost, but not too much, so we can't get to sleep later at night, so the matcha is perfect for that. And Ellie, you said you were going to take the Mushroom Magic cocoa, right?
Yes, I'm having the cocoa, but I did add a shot of espresso, so it's for me. So I'm kind of on the opposite end. I would love to be awake for the next 8 hours and energetic and focused.
Yeah. Awesome.
So let me mix ours up while we get the conversation started. So,
great.
Ellie, if you kind of want
to,
nice.
If you want to describe a little bit about how you got started on this journey and what your research ended up being about.
Yeah, definitely. If anyone listened to the first podcast I was on as a science director, we talked about this a little bit. But going to grad school, my interest was definitely botanicals, but I didn't really know what aspect of botanicals. And I would say I stumbled into dietary supplements more by my interest of mass spectrometry, which is the use of large chemical data. So I was really interested in how we can look at the individual compounds in plants. I think it's really fascinating that we have the technology to be able to look at individual compounds and say, what compounds are there and how much of them are there and why, and how can we manipulate plants to have more of those compounds that benefit us, but then also how do we respect plants and what they're already giving to the environment. And all of that came together in studying plant chemistry. So my interest was more in the plant chemistry side before the supplement side, but then once I started learning about more dietary supplements and how many are botanical focused, I realized we can combine the two and really improve the marketplace. And with mass spec, you're getting a lot of data, and we'll talk more about the details of that. But the best way to handle that data is through machine learning. So you're looking at so much data at one time, there's no way you'd be able to comprehend it all without using large algorithms and using statistical tools to take, thousands of compounds and find the important compounds that you're interested in. So through that and through my interest in plant chemistry, I also stumbled into statistics and machine learning and things I never thought I would love so much. But it's kind of once I started getting into the coding of it and the sitting down with the data and really getting to manipulate the code, manipulate what the model is doing for me, I realized how powerful of a tool it is. And that might not give so much explanation of to what I did with it, but that's how I got into it.
That's great. And it's great hearing that there are people on this planet who enjoy statistics, because I remember from college that I had to do a lot of statistics courses too, because it's also a big part. It's a big part of any scientific endeavor.
Mm-hmm.
But that was always my least favorite thing. I always thought in school, I'm going to focus on the other side. I'll let someone else focus on the statistics. But at the end of the day, you can never really escape statistics. You got to understand it and manipulate it. It's a powerful tool.
Definitely. And I think there's a big difference between kind of like your basic statistics you learn in college of the study design and t-tests and more of the basic, how do you tell things are different? And then going into things like machine learning, which is using large algorithms. And it's just a different kind of statistics.
Yeah.
That it's more like a puzzle.
That
Putting
sounds
together
more
a puzzle to get an answer that you don't know yet.
Yeah.
It's really
cool.
1
second. I'm just going to shake this up. It's going to make a bit of a noise. And we are drinking, as you can probably see based on the color, the Baller edition, because the normal one is not nearly this green. That's really a result of the matcha that's in here. And just a real quick story about the matcha in here. We recently picked up some matcha from a fancy coffee shop in town. We really like their beans. We thought their matcha must be good too. We want to try making some matcha lattes and stuff like that. And we got home and we were quite disappointed because the pure matcha powder didn't taste as good as this, which also includes mushrooms. And it's because we have really, really high quality matcha in there. So we have our matcha source now because we get that nice matcha taste and the functional mushrooms that are in here. So
yes, if you're a matcha tea lover and you think that the coffee shop that you like to go to has the top of the line, just try the Mushroom Magic Matcha Baller and you might be pleasantly surprised.
Even the smell, like if I'm smelling the bag from the fancy coffee shop, the pure matcha, it doesn't smell nearly as pronounced and fresh and floral as it does when it's already mixed up here.
Yeah.
And the concentration is much lower.
And even without added sugar or sweetness, the matcha in the Mushroom Magic Matcha Baller does have just this natural grassy sweetness to it, which is really, really nice. And then if you use some high quality milk to make yourself a matcha latte, it's like perfect.
It's really nice. Can you give me your glass?
It's really good hot and iced.
Yes.
So,
I particularly like the Baller edition cold because I feel like some of those floral tastes are a little bit better preserved, but it does taste a lot more classic when you do it warm, which is also really nice.
Cheers.
All right, back to the real conversation. Just a quick mushroom magic break.
Yes.
It's become a really fun drink actually on the podcast. I feel like this is almost a mainstay, and I do want to give you credit for that because you were the first one to bring up, let's do a stack of Cognans and Mushroom Magic Baller, and that worked really, really well. And then on our last podcast with one of the people from our scientific advisory board,
Carrie Hughes, in case you're curious, we will link the podcast in the description.
When we asked her what she wanted to take, she basically said the same thing. She wanted to take Cognans and Arinimax. And Arinimax is also in the Mushroom Magic Baller. So we had two experiences where we did Cognans and some sort of lion's mane mushroom-based drink. I do think that the Cognans with the Mushroom Magic Baller edition that we did with you, that was the most pronounced effect. Really fantastic.
it's crazy, yeah. And that, to steer more away from the topic we're actually talking about, I've now been working with Nootropics Depot and taking the supplements for over 3 months consistently. So I've been taking Cognance very consistently, and either they're having a Arinimax or one of the Mushroom Baller Edition drinks daily, and I can feel really strong changes in the way my brain is functioning day to day. I typically have kind of 1,000 thoughts going on at a time, and probably like something spinning in my head too. It's just like a lot going on up there. But ever since having that combination daily, I can tell things have slowed down in my brain. I'm able to multitask more effectively. And that's really the only thing I've changed, is adding those supplements in. And so I can tell immediate effects from that combination as well as long-term actual substantial changes in the way my brain feels like it's functioning. So it's nice to see that it's more than just an immediate effect.
Absolutely.
Yeah.
And that's, of course, something that the viewers will miss because you only see the acute effects here on the podcast, but this also builds up. So if you drink the Baller edition every single day, you are definitely going to get more increases in memory, mood, things like that. Those kind of effects, if you respond to it right away, they will continue to improve. And sometimes even if the effects aren't that strong right away, they will increase in intensity over time. But I think it would be pretty hard to miss the effects of something like the Baller editions, because those are surprisingly potent.
Yeah. When I first tried the Mushroom Magic Matcha, I was amazed. And this was just the normal version, not the Baller edition. Because I had never felt a mushroom product from Nootropics Depot have such a pronounced effect right away. So I was like a super fan right from the start. And the Baller is a big, big step up. And I remember you had tried it first and you were saying, oh, it's pretty intense. Like the higher end, you know, the more premium mushrooms make it really stimulating and really energizing. But I don't find that it's overstimulating at all. And I'm similar to you. I have like a million thoughts going on in my head, so many projects, so many ideas. I wake up and I'm like, okay, like I'm going to climb the mountain today. And then I drink the matcha and I'm like, someone said this, I don't remember who it was. If you look at the top of the mountain, you'll never get there. If you just look where you're walking, you will get to the top of the mountain eventually. It helps me do that. It helps me just stay focused on the task at hand.
That's like the perfect explanation of how it feels. It's like, okay, I still have all of the things I want to do and the ideas, but I can actually focus on the ideas.
Yeah.
Yeah.
Speaking of which, let's get back to the topic at hand. And one thing you mentioned that I really like is that you mentioned that the statistics that you were working with are like a puzzle. And this puzzle idea feels a lot more lighthearted than my idea of a statistics class. And for a little bit of background, I struggled so hard with math when I was in high school that I chose not to take math in my senior year of high school because I studied music and I knew that they wouldn't care about my math scores at all. So I didn't get anywhere near statistics and I don't know if I ever will. But hearing you describe it like a puzzle, it's really clear too that you really enjoy this and that you have an understanding of it which allows you to see it in a different way. So let's talk a little bit about the machine learning, because machine learning just as a concept is something I'm not so familiar with, but this is your whole study. So can you give us just a general idea of like what is machine learning and then what did you do specifically within that?
Right. So machine learning is a pretty broad category. It's kind of any situation where you're providing an algorithm or a computer data, and then you build the algorithm so it can find patterns within that data. So I'm not going to get too deep into the actual statistical terms because, I mean, it'll bore even myself. But essentially, you want to take your big data and see patterns that we would never be able to see. And then you can do things like, based on these patterns from this previous data, what do you think this new data is? So that would be more of a classification problem. So in terms of botanicals and my research, I would say, based on all of these different types of basil, these are their chemical profiles. This is all of the compounds in 30 different kinds of basil. Based on that information, I have this new basil that I ordered online. What kind do you think it is? So maybe taking a step back to talk about the basil side of that before we get more into the discussion of the algorithms. Basil sounds like a really straightforward plant. You know, you grow basil in your garden. There's holy basil, that's what we have as teas. But there's actually over 60 different species of basil, which I don't think most people know. And while a lot of those species will have the same, you know, effects when we take it as a supplement, they do have different chemical profiles. And so it was a really good model species because I could grow a bunch of varieties. I grew 30 different varieties of basil. And I could look at their chemistry but it wasn't hard to grow. So I was able to grow it in a greenhouse, manipulate some environmental conditions, but still get varied chemical profiles. So I used that variation between different varieties and for different species of basil to build predictive models. So those models, as I said, use their chemical profiles to say, okay, this new sample of basil looks the most like sweet basil, or it looks the most like Krishna, holy basil. And so I could say, this basil that is being marketed as a holy basil tincture is actually just sweet basil that you would grow in your garden. Mm. And that's based off of similarities and differences in the chemical profiles. And while for basil that might not seem so important, you're not going to have adverse effects from taking the wrong kind of basil. When you apply those algorithms to, say, plants like terkestrone, or if we were looking at the difference between two different kinds of ginseng, or plants that are more expensive, that's when those algorithms can have a bigger role. So that we could say, we know based on the chemical profiles what this is or what this is not.
So is this would be a continuation of HBTLC, doing things that HBTLC couldn't do, or is it more like a companion? You would still need to use HBTLC? I read through your study a bit, which, by the way, for those of you interested, we will link it, of course, in the description, because Ellie is really a published researcher as well, because your article got published in Nature, right?
It was actually in analytical and bioanalytical chemistry.
Okay, nice.
Pretty far away from Nature, but pretty big in our specific field.
Yes, yeah.
So, and then I have a review for anyone interested in learning more about the actual kinds of algorithms that we use and the different kinds of models. I have a review paper that talks about every kind of model being used for authentication of botanicals, how it's been used, and kind of some future directions. And yeah, we would definitely link that below. And that gets more into the actual details of the statistics. And then, the paper with the basil authentication is a published article as well. And that one, ultimately, we found that doing this kind of machine learning is actually not a sufficient way to authenticate botanicals. So far I've said, we can classify, but it is just an idea right now. This is a new field. A lot of people are trying to use this for authenticating botanicals, but it's not at the point yet where the algorithms are sophisticated enough to do the prediction accurately. Mm-hmm. And in my specific study, I found that there was a lack of variation when you grew all the plants in the greenhouse. Mm-hmm. So we weren't encompassing different types of drying, different altitudes of growing the basil, different soil conditions, different pests and pathogens. And all of these different things have an impact on a plant's chemistry. And without encompassing all of that variation, you're never going to have a model that accurately represents products that are on the market. Mm-hmm. And that's the biggest limitation right now, is having enough authentic material to build models that are actually representative of the marketplace. Mm.
Which I think brings up a broad issue that we've touched on a lot on this podcast, is that a lot of what's claimed in the supplement industry isn't actually real. You might be buying Ocimum sanctum, or what's the other Latin name for holy basil?
Tiniflorum, Ocimum tiniflorum.
Yeah.
Ocimum sanctum is actually the same thing. They're just different names for the same thing.
Which one actually is correct, or are they both correct?
I think sanctum is used more. Okay. But they're both correct, and they're both the same species. And then there's also Ocimum gratiseum is the other holy basil species that I studied. And then sweet basil is Basilium.
Okay. And that's the one for those curious, that's what we eat mostly, right? The sweet basil?
Yeah, if you're making pesto, that's going to be what you're using.
Nice.
What I found really interesting about the study is that, from what I could gather, the method worked for the plants that you grew, but then not for the commercial samples, which brought up a lot of ideas in my head, and things we have talked about in this podcast a lot, is that when we dry, we lose things. When we extract, we lose things. And I thought one really interesting thing in your sample prep is that you immediately put everything in liquid nitrogen, right?
Right.
And Erica used to work at a restaurant in New York called Sambar, which is being
Momofuku Sambar.
Which is being run by David Chang, and connected to that there was a kind of a molecular cocktail bar being run by a guy called Dave Arnold.
It was called Booker and Dax. It was amazing, but it no longer exists, sadly.
And they had rotovapes to make things, and they had I'm blanking
liquid
on
nitrogen.
They did a lot with liquid nitrogen, and this is one of the things he was doing.
They use siphons a lot to create different foams and things.
Yeah, and it was the centrifuge.
Yeah.
Centrifuge they used to clarify a lot of different things. So they would take lime juice, put it in a centrifuge, and then take the fraction out that was completely clear, and then you can carbonate that. You could do all sorts of crazy things with that. But one really interesting thing, and maybe the most interesting cocktail I've ever tasted in my life, was a technique he came up with, which was called cryo-muddling, where he would take fresh basil leaves and he would put them in a shaker tin and then put liquid nitrogen in the shaker tin and take a muddler and muddle it all and then put high-proof spirits in it. And his thinking was that there's an enzyme in basil that degrades the positive volatile aromatics. So if you take a leaf of basil and you crush it up, you break the cell walls, then that enzyme gets released and it degrades the sensory attributes that you would get if you just smell a basil leaf fresh. So he thought at really low temperatures that enzyme can't get activated, and then you break it up, and then all of the cell walls are broken, so that enzyme is easily accessible by the alcohol that you then add to it, and the high-proof alcohol in the research he did deactivates that enzyme and keeps all of those volatiles there. And it really smelled like the most basil-y thing I have ever smelled. So going back into your study, I thought it was interesting that you're basically doing a very similar process.
That's almost the exact prep that I would use in the lab. Yeah, we would flash freeze things in liquid nitrogen, just because that really does keep the exact chemical profile of the time you harvest it. And we harvested all of the leaves of the plant. For any botanical scientists listening to this, that's an important part, is we weren't just looking at the basil leaves, but the base leaves. We were looking at all of the leaves, and we harvested right before flowering. So it was very consistent. And so all of the chemical profiles weren't swayed by time. So we wouldn't be seeing differences in harvest time or the type of leaf. And when you flash freeze it, it really contains those compounds. So you're looking at that exact time, what was that chemistry like? Which is great for the greenhouse study, because we were able to use machine learning to differentiate between the different species of basil and the varieties of basil using machine learning. But because we had such high control of those conditions, we weren't able to model anything outside of the greenhouse.
And I think that goes back to with drying we lose things, and then a lot of the things you probably tested were also extracts. I think I read you had some whole leaf stuff, but also some extracts. And in those extracts, you always lose something as well. And a really good way of highlighting this is if you make a cup of coffee, you end up throwing away the coffee grounds. You get an extract and you're throwing away a lot of it, and that's how all extracts work. And that stuff that you're throwing away is probably important markers as well, and bioactives.
Exactly.
So we are more and more now playing around with non-extracted plants, because if you grow them to a very high degree and you can manipulate maybe the cultivars, the growing conditions, you can get very high amounts of bioactives without having to do any sort of extraction and concentration, which then has a higher degree of bioactive diversity. And I think that's also where some of your research comes into play, maybe being able to deal with a higher amount of bioactive.
Before we
Yes,
get
exactly.
too deep into this direction, I do want to mention something fun because you mentioned that the prep process for the basil, the way that Emil had described it, it was almost exactly like what you had done in the lab. For anybody who's curious about trying to make some of these cocktails at home or just reading about it, there is a book that Dave Arnold wrote, and it's called Liquid Intelligence, and we can also link that below for anyone who is like a cocktail nerd, a nerd in general, curious about this kind of process and curious to read about it. It's a really, really fun book. So I just wanted to mention that before we move on.
Yeah, if you ever thought science is boring, which it isn't, as Ellie and I can attest to, but if you thought it was boring, go read Liquid Intelligence and it should change your mind, because he can do a lot of fun stuff with science. And we do a lot of fun stuff with science. If it wasn't for science, you wouldn't be able to have something like the Mushroom Magic Baller edition to drink, and that's pretty fun in my opinion.
Totally.
You mentioned HPTLC, and that is our most standard way of authenticating botanicals and making sure that they are the plant that they say they are. A problem with HPTLC, and this is not just my speculation, it's something talked about in the industry a lot, is the lack of reference materials. So with HPTLC, you're looking at one botanical reference material. So we'd be looking at one basil that was authenticated to be basil. But like I mentioned, there's over 60 species of basil. Within that, each one has different cultivars, and it's a really widely kind of differentiated plant because we want it for different uses, different smells, different tastes. So that one botanical reference material is not going to encompass all of the chemical variation. So with my study, we had the problem of what we grew in the greenhouse was not representative of products that you can actually buy. But with HPTLC, we see the same thing. We see that with one botanical reference material, it's rare that you're getting all of the chemical variation that you can see for that botanical species. So no approach at this point is perfect. And my prediction is that there's going to be machine learning models soon that not only uses all of the information from metabolomics or mass spec, but it combines that information with HPTLC data and with DNA barcoding. And soon we'll have algorithms that can take all of the different information and combine it into one answer. So it really is no one answer fits all. It's you need a bunch of different information to find your answer. And we're not there yet, but that's kind of my prediction of how we'll be able to use machine learning for botanical authentication in the future. And it really highlights, and Emil, you can definitely speak about this maybe more than I can, the diversity of plants. And we have to remember we're working with plants. We're not working with something simple, even though plants, for a lot of researchers, people seem to look at plants as something simple. They're actually so incredibly complex, and they can't tell us what they are, and that makes it really hard to research. It's a fun problem, but it's a problem that we face.
Yeah. And for that diversity, if you can actually see the plant in real life and it's growing, and especially if it's flowering, then there are a lot of more visual indicators, and a botanist can go in and go, that's maybe Ocimum sanctum, this one is Ocimum basilicum, just based on how they look. But I also found it interesting in your paper you stated that sometimes you have purple varieties of sweet basil that look very similar to holy basil, so then they're a lot harder to differentiate already. And then we need analytical tools.
We actually had to use seed morphology for our authentication. So, to confirm, you can order seeds online from places, and that's what we did. But until the plant grows, you don't really know what it is. Maybe the seeds got confused or they were incorrectly identified. So we did morphological identification to make sure how we were classifying them in our models was correct. And the best option was to use the seeds for the morphology, just because the overlapping species and species that are so similarly related are going to look the same. So even just using traditional morphology has its complications as well.
And what is morphology?
That's exactly what Emil just said. So looking at the way the plant looks.
I see.
Things
Okay.
like flowers and leaf shape and even things like smell can kind of be classified as morphology.
Cool.
And from those seeds, how many of those seeds were not what the seed sellers claimed to be? Or were they all pretty much what it was?
They were all basil. A lot of what we saw was the two holy basil species that I use, Osmum gratisium and Tiniflorum, were actually the sweet basil, which is what you would expect. It's a lot easier to grow sweet basil. And since it does have so many different varieties, they can get misclassified pretty easily. So I'm glad we checked.
Yeah, that's good. And interesting to know, too, that the problem of things not being what they are extend beyond the supplement industry. If you're buying some seeds to add to your garden and you want to really grow some holy basil and you order seeds from somewhere and you just get a normal basil that maybe has slightly more eugenol content, and you might not even notice the difference because
Exactly.
yeah, but it's not what it is. So if you're a real nerd and you really want to know what it is, check your seeds under a microscope or whatever it is. It's the same in the mycology world. There's a lot of spore sellers online, and sometimes you really have to grow those spores to see what the mushroom is based on its morphology. Okay, yeah, that looks like lion's mane, but you can also look at those spores underneath a microscope and then go, yeah, that matches in the research. This is how a Heritium erinaceus spore needs to look. I actually got Heritium erinaceus spores and not Heritium coralloides spores because those look different. They're different sizes, different morphology, which is that's a really interesting technique because it's pretty low tech actually.
And there are machine learning algorithms for microscopy. So a subset of machine learning is using image patterns. And so people are coming up with algorithms for, you take a bunch of images of different spores or seeds or plant cells and then put them in the algorithm, and then based off of the microscopy data, it'll predict what species it is. So, and it's kind of the same thing. It's the scaled down micro version of things like iNaturalist, which uses machine learning, AI image softwares to take a picture of a plant that you take on your walk and pair it with, thousands of pictures of other plants to figure out what it is.
In researching a little bit for this podcast, I also came across a technique called hyperspectral imaging. Have you looked into that at all?
I haven't.
It seems really cool because it's really just kind of a normal camera, and you can take a picture of the plant while it's growing. So this specific research I was looking at was for the hemp and cannabis industry. So they could just go a growing live plant, and without having to do anything, they take a picture, and then it's a picture in 3 dimensions. So the first 2 dimensions are just normal, color and depth, how we would perceive a picture, but then that third dimension is spectral data, that's like near data. And with machine learning, they're able to determine if it has a low amount of THC and a high amount of CBD, so they can say that's like type 2 hemp product, which has low THC, higher CBD, or it's type 1, which I believe is high THC, lower CBD, and then type 3, which is THC below 0.3% and then CBD, so you can sell it as hemp in the US. But they can just determine it by taking pictures. A very special camera, by the way. I was looking them up because I thought, I kind of want this. Like, maybe I can start taking pictures of stuff. They're about $75,000 for a really, really basic thing. So that's not there yet. But I do think in the future, with AI getting so much better and consumer AI getting so much better, and we'll touch on that a little bit too, because I think it's interesting to talk about some of the new updates on ChatGPT and Perplexity, some of the AI tools that you already can start to use at home without having to do a PhD about it, because the stuff Ellie is doing, you can't just do at home, I think. It's quite complex.
I don't know. I do think I think anyone could learn how to do machine learning. It's the tools are there. I basically taught myself. It's, you have to know how to Google things like how do I fix this error? And that's kind of what you do all day long is, okay, now how do I fix this other error? But I think it's something anyone can learn how to do. It's just having, the funding and the guidance to do the research projects is super important. And that's definitely something we're seeing in, the most recent weeks is funding is very important. And this research and supplement safety and botanical safety is incredibly important to us. And we want it to be important to consumers. And the research has to be continued to be able to advance safety.
Mm.
Yeah.
Do you think 20 years from now, 10 years from now, maybe even with how rapidly AI is progressing and how rapidly some of these technologies are progressing, that you could maybe have a near chip on a camera phone even, and you can just take a random powder, scan it, and it tells you what it is. Do you think we can come that far, or is that kind of a fantasy dream?
I think it's possible. I think botanicals are so complex. It's going to be a long time before we're at that point. Mm-hmm. And there's so many different botanicals. And I think it took, millions of years for botanicals to evolve. And AI is evolving very, 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 very rapidly. But I don't think it's ever going to catch up to the complexity that botanicals have. Yeah. Which is great for us. So, our jobs aren't going anywhere. Yeah. But I think it's going to be more of a companion tool for people who understand the botanicals themselves.
I just find it fascinating these days, and I think talked about this a little bit too, the Purple Scientific, handheld, Bluetooth connected. I think it's also using NIR technology to test hemp and cannabis flower, and I recently saw another one called the Neospectra. And these things are within a consumer budget. If you're a real hobbyist, you can already buy a Purple Scientific for $2,000, and if you're growing hemp at home, you could test it yourself and get a rough idea of what the potency is like, or actually quite a precise idea of what the potency is like based on some of their data. And with the Neospectra, it can even test for terpenes, which seems almost unrealistic to me, but they've also quantified it against more established methods. So seeing these things, a Neospectra is costing $5,000. They even have them now where you can attach them to a drone and just fly over your crop, and that can already get data. So some of and it's all with machine learning, and apparently it can kind of fly over and see, oh, this plant is a little bit low in magnesium because it's a little bit less green. You need to add a little bit more magnesium to your water, and then you'll get better crops. Or it flies through and it can already detect pests, but then on top of that it can do some level of potency analysis on a drone flying by, which seems like Harry Potter-esque to me.
Right.
But it's there.
How?
Yeah.
It's amazing. Yeah. And this is all way, way beyond my experience or really level of understanding, at least the systems themselves and the technology. But from our last podcast, the podcast with Carrie, we talked a lot about just our human experience with plants and the plants that are in our garden and the plants that we interact with on more of a daily basis. For us, for botanicals, that's often coffee or tea or matcha. Or if you wear perfume, there's botanicals in your perfume. If it's actually coming from real plants, maybe it's synthetics. But it's interesting to learn about this technology, which is basically trying to mimic that of a very experienced farmer, that of a very experienced grower. Because when we first started this podcast, we started talking about tart cherry. I think that was actually our very first podcast episode. And we discussed the fact that the color of the tart cherry also indicates its anthocyanin content. And previous to this podcast, I had no concept that the color of a fruit, vegetable, plant could have some level of significance in terms of its nutritional value or its effectiveness as a supplement. But we talked about this, and we talked about the fact that the pH level determines the actual shade of red or purple, and that the red and purple is also somewhat of a protective mechanism, and it also helps us in a protective way when we ingest this, and it's the same color property, it's the same compound that's also present in purple yams and in blueberries and in grapes and all of these foods that are purple, red, even strawberries, which has a different pH level. So there's this amazing technology that's being developed to do this kind of work, this analytical work really quickly, let's say over a field of plants. And it's so cool, and it's also fascinating that this work is being done by machines, and people have been doing this work for all of time. Like, the machine learning is trying to catch up to, like you said, the complexity of the plants itself, but also the complexity of our knowledge and our experience of the plants as human beings. And I think it's refreshing to hear you say that the machines are never going to catch up to the complexity of the botanicals, because it gives me a bit of hope in just reality of like the stuff that's actually growing in the ground, that the machines can be really smart, but eventually what we all kind of, the conclusion we come to, or at least the conclusion I come to, is we all have to eat, and hopefully we can smell and taste and experience the things that we eat. And then in the case of supplements, we experience the benefits of them. No matter how smart the machines get, as human beings, like we're learning and developing this technology to be able to better understand it, but it still doesn't mean that robots are going to take over the world, at least not quite yet.
Not quite yet.
But on the other hand, it also indicates how dumb human beings actually are. And I think we sometimes think that we're super smart and we know everything better, but a being is not able 9 times out of 10 to differentiate between a purple Ocimum basilicum plant and a similar looking Ocimum sanctum plant, even though they're two completely distinct species. And you would think you could just look at those and get the seeds from them, and the seed seller could sell you the correct seeds, but they can't. So having some degree of technological help, I think, is quite critical also if we want to develop better products. And I do think, just to bring it back to the machine learning, a really interesting thing you said is that we can use kind of our eyes and our noses as analytical tools. And a lot of these analytical tools that we're talking about are actually working based on the spectrums, the light spectrums that are coming off of these things. So what I was talking about with NIR testing, like these purple scientific or NEO spectra, it's really shining kind of a beam of light at your sample, and how that beam refracts back and goes into the analytical machine, and then it gets all sorts of mathematical equations happen, and then you can go, based on how the light is reflecting back, this is what it is. And I think even in an HPLC, UPLC, there is some element of light and how that's being interpreted, right?
Yeah, it depends on the instrument you're using. So in our lab, we most commonly use a UV coupled to the mass spec. So that's looking at how different wavelengths of UV light is being absorbed, which obviously we can't see that. But it is still based on the basic principles of light hits a sample, light gets refracted, and then you can compare how is that light refracted. And that gives us the visual information. So it's very similar.
Yeah. And like Erica was saying, a tart cherry is red because of its anthocyanin content. The redder it is, you could assume the more anthocyanins are present. And that's really based on the color. And I think a really simple one is any plant that's green, you could very confidently say contains chlorophyll, because the green color is coming from chlorophyll. Plants need chlorophyll. We see chlorophyll as green. So that's a really simple way of doing it. And then analytical tools just take that to the next level and can interpret things that are otherwise invisible to us.
So speaking of green, let's talk a little bit more about basil. I'm also really excited that you mentioned that you grew 30 different varieties of basil. And just a short little anecdote, we bought some herbs last year from like a specific herb grower, and I had never encountered so many different varieties of basil there. There were a bunch of really cool, really strange, brand new varieties I'd never experienced, some with like sort of exotic fruit scents as well, and
Wow.
and we tasted them too. We would just like take a little leaf and we would taste them, and some of them tasted like the name or like the sort of exotic fruit that it might taste like. But some of them just tasted like plain basil. But I had no idea that there were just so many different kinds, and this was the case for basil, but also a lot of the other herbs as well. So this what you said just really reminded me of that experience. And how many different varieties of basil did you say that there are?
There's over 60 species,
Species.
and then within each species, there's different cultivars. So cultivars are things that humans have kind of changed the chemical profile of. So, you know, sweet basil, there's purple basil, and then there's your common sweet basil. The cultivar is plant breeders choosing which traits they want to optimize and cultivating it to be different. But then there's different natural varieties. So, say the same species of basil grew in two different environments, and to survive in those different environments develop different chemical features. That would be a natural variety. So because there's so many species of basil growing in so many different parts of the world, there's a lot of different natural varieties, but it's also something we love to eat. So there's a lot of cultivars as well. And that just makes the taxonomy and the variation very, very complex. And it's like that with most herbs.
Especially herbs that are really important to us or financially important. I think, again, to bring up cannabis, that's a really interesting species because within that species you probably have tens of thousands of cultivars at this point because it's been financially lucrative to breed the plant, to have different expressions, but it's all the same plant. And if you look at the chemistry, it's wildly different. If you smell it, it's wildly different. If you consume it, the effects are wildly different, but it's all the same species. And there's just hundreds, thousands of different varieties, very distinct. And I think also...
That makes,
go ahead, Ellie.
well, it makes ID hard for cannabis, pretty straightforward because those tools they are very well funded in the ability to develop tools. But for things like smaller plants that, you know, sustanch or something that only a few companies are selling and there's only a few sources of it, that makes it harder to identify because of that type of variation. And how do we know, is it the same species with just broad variation or are they actually different species?
Yeah. And then, of course, the issue of botanical reference materials comes up because at the end of the day, you do oftentimes have to trust someone to send you a botanical reference material and you have to get it verified, but how many botanists in the world can distinguish between Cystanche tubulosa and Cystanche deserticola? I'm actually not sure how different those plants are, and I wouldn't be surprised if 99.99% of botanists on the world have never even heard of Cystanche.
Exactly. I have a Ph.D. in plant biology and I hadn't heard of half of the plants we talk about. So it's a cool thing to learn about, but definitely a little overwhelming.
Yeah, for sure.
Which is a compliment for us too, because it means we've been trying to specialize in novel niche plants that not everyone is dealing in, which, it's become quite overwhelming for, I think, all of us. But we're really on a mission to discover all of the different types of plants that are out there. And it would also be really interesting to get into different cultivars, especially because we've been doing that for our Lion's Mane project. We have many different cultivars of Hericium erinaceus, and then also other Herician species, and we're seeing big chemical differences between them. And also just looking at it grow, it's different. The mycelium grows differently, different biomass, growth speeds, all of that can be optimized, and there's a lot to explore.
I do think the topic of how complex this all is like a good segment into how we've been using AI tools. So AI is different than machine learning, and I'm sure there are lots of podcasts I can talk about that in better detail than we can, but we've been using AI a lot as a tool in product development, and it's definitely a shift away from what my thesis research was, but I think it would be fun to talk about how we've been using things like Perplexity and ChatGPT to help us develop new products and understand the plants a little more.
Absolutely. Do you want to start, or should I start on that topic?
I think you should start on that
Okay.
one, since you're more of our product guy.
I think we both have a lot to say on this. I'll say up until this year, I thought AI was unusable. We've been attempting to use it. A lot of my colleagues have been over the moon for it for many years, and I've not gotten it until about 6 months ago or so, when Perplexity came out. Because I think ChatGPT is just super general. I know more than ChatGPT. I'm really not learning much from ChatGPT. Like, I could learn ChatGPT a thing or two. It's not picking out novel, interesting, in-depth research. But when Perplexity came out and we signed up for the Pro account, which has some deeper analysis, I'm not exactly sure about what kind of models it's running now, but when we first started using it, it was good, and over the last couple of months, it's really, really improved. And we can ask it questions, and it will pull up summaries based on what it thinks is important, and it will also link to the research. So we can go into the research and then read the research, come back and ask Perplexity a more refined question. And through this, I think personally, I've been able to do about a month's worth of research in an afternoon. Because
Yes.
I'm not having to go and parse through a bunch of completely useless studies. Because oftentimes you read stuff and just nothing positive comes out of it, and maybe you find another link and then you're hours further into a rabbit hole. Now, it can do that first parsing of information for me, and it can reveal things that I didn't know at all, and it would have taken a really, really long time through a lot of research on NCBI to find this, or talking to researchers. Because for me, it almost really feels like I'm having a exciting chat with someone, and it keeps bringing things back to me that give me something to think about. And when I've really thought about it and processed it, I can go back to Perplexity and ask it another question, and just being able to find the research every single time because it links to it is really beneficial, because a lot of these AI tools aren't really that smart at all. Sometimes ChatGPT would pull stuff up that I knew was false, and there is no way to verify it because you can't actually click on the research where ChatGPT pulled it from. At least not anymore. You used to be able to, and it didn't work so well, and then they stopped doing it. I'm not totally sure why, but it just doesn't really work all too well. Perplexity is really different, and Perplexity is so powerful that we've even been plugging some of our analytical data into it, and figuring out maybe what other people are doing with their analytical methods or extraction methods. So it's that powerful that we can go, okay, if we want to have the best Tonga extract on the market, what do we have to do? And we already had some really good ideas about that, and then we plugged those ideas into Perplexity, and then Perplexity spat out a bunch of other ideas that made all of us go, wait a second, this is really
Never would have
unique.
thought of that.
Yeah. And for me, a really interesting one that Perplexity helped me with a lot was sulforaphane, and how glucoraphanin, the glycosylated, the sugar group containing version of sulforaphane, gets converted into sulforaphane. And that's happening through an enzyme called myrosinase. But we've always looked at myrosinase as you just add myrosinase to glucoraphanin and then it converts to sulforaphane and we're good. But that doesn't seem to be so simple, and Perplexity was really instrumental in teaching us about different factors that I'm not going to say here because we're actively working on a project with this data, so I don't want to give it away for others to use. But soon enough we can talk about it in more detail. But that's been a really powerful one for me to do months worth of work in a single week, and I think for a lot of us. So the thing that I'm most excited about is it accelerating our research and being able to do things that might take us 10 years in a year, things that might take us 50 years in 5 years. And I'm excited to see not just how that will change our product development, but just the world in general, because we have so much more access. And some of these new AI models that are coming out, like the DeepSeek, apparently they have a research function that's really targeted towards people who are researchers or people like myself. I'm not a researcher, but I read research all day long, so I'm quite well versed in a lot of the topics that are being discussed. And then one last thing I wanted to touch on with Perplexity is that I've also been using it to find new bioactives that we might want to look at in certain plant species. So I had recently from an order from an ethnobotanical website as an inspiration from our episode with Carrie, who's an ethnobotanist. I thought I need to play around a little bit with some of this ethnobotanical stuff and do some research there, and I found one product that I really liked, and I was really curious what in that plant, which the plant is called Canela de Velho, which I'm forgetting the name now, but it's a plant in the Amazon. It hasn't been used very much outside of traditional cultures there, it seems like, so there's very little research on some of the bioactives as well. I had to do a lot of digging, and it took me quite a while, and then I thought, hey, I can just use Perplexity. So I asked Perplexity, tell me all of the bioactives that are in this plant, and I put in the Latin name, and it really did pull up a huge list. And from that, I was able to go to various research studies that I wasn't able to find previously, and they had big lists of all of the bioactives in there. And then I took some of those bioactives that seemed really interesting, and then I went back into Perplexity and I said, Perplexity, what does this bioactive do? What are the pharmacokinetics like? What are the pharmacodynamics like? And it spits out really good pharmacokinetic data if it's available, and pharmacodynamic data. And based on that and some of the analgesic effects that are mentioned and that I noticed as well, I was able to find the compound pretty quickly, the amarinds. And that then I asked Perplexity again, what other plants are the amarinds in? And then it pulled up a bunch of other plants that all seem to have really unique analgesic effects. And then from there, I looked at some of those plants and looked into some of their other bioactives, and it was this huge rabbit hole that I went on, discovering new bioactives. And Ellie has something that she's worked on with her thesis as well on that she can touch on a little bit later. But I think that's a really interesting topic to explore, because there are a lot of unknown bioactives and a lot of plants that aren't well discovered, and I think a lot of those bioactives have huge potential for us, also for the pharmaceutical industry, of course, because a lot of pharmaceutical drugs are actually derivatives of natural products. Sometimes they even are natural products, like morphine, one of the most used surgery drugs on the planet probably, is a direct extract from the opium poppy. There's no synthetic anything happening with that. They're actually growing poppies and extracting morphine from those poppies and still using it in modern day healthcare. And I know there's a few other compounds like that. There's also a compound that's being used in cancer treatment, I think, that is in the plant is called buxus in the Netherlands. I forget what it, but it's like a common ornamental plant that a lot of people have. And it contains a really potent anti-cancer compound that people will send in their clippings of this plant to research institutes, and they'll extract that compound out of it because they don't have the means to synthesize this very complex compound. And there is a good commercially available source for it that's just growing in people's gardens, and people routinely have to cut that plant into a nice shape, and then you have a bunch of material to give. But with some of this machine learning, I think we can really accelerate our prospecting of a bunch of different botanicals to find interesting lead compounds for pharmaceutical development, and then also for us, for the supplement industry.
Definitely.
But how do you use Perplexity? I'm quite curious how you're using it in your day-to-day life now.
Well, it's funny to hear how you use it and how I use it is it's very similar, but also a little different, even though we're typically on the same meetings, probably using it on the same topic at the same time. Because I'm typically more on the, okay, here's the analytical data that I have about this plant, and here's what people have reported feeling out of this analytical data, what peaks are interesting, or what methods should we be using to analyze it. And I think that kind of feeds into the AI algorithms can only operate on the information they have. And so you mentioned something about if the pharmacokinetics are available, it can tell you about it. But the research still has to be done to see, what are those compounds doing and what are the compounds. And that kind of feeds into one of my other projects I worked on as a grad student, which was using machine learning to discover new compounds. So while AI can tell you previous research has said all of these compounds are in this plant, I was kind of doing the research to say these compounds are in this plant so that we could publish that. And with my research, I was doing basil for this project as well. Most of the compounds in basil are pretty well known, which had its benefits for me. But when we're discovering new bioactive compounds, the traditional way to do it is to say, I have this plant, it's got all these compounds, and I feel this effect. I'm going to take all of those compounds, fractionate them. So now instead of looking at thousands of compounds, I'm looking at hundreds of compounds. Do we still see that effect? If yes, do it again, and do it again, and do it again until you have one compound. That takes a long time. And you also lose kind of the interaction of different compounds. And every time you manipulate the plant material, you're losing compounds just from extracting or messing with the material. And so I used machine learning to say, okay, I have these thousands of compounds. I have these plants that have different levels of activity. So I was seeing how the basil plants affected how well cells grew in a petri dish, essentially. And then I gave the information about the cell viability and all of the chemical compounds in the different samples and said, what compounds are different between the ones that are good for the cells and are bad for the cells? And I was able to use machine learning models to find a single compound that was most likely causing the effect between the samples.
Wow.
And so that saved months and months and months of research time and materials and experiments not working because I could do it all at once. And I was able to find a compound that was different between the active and inactive samples. And luckily for me, since I was using basil, it was already a known compound. So I could just order the standard and confirm the predicted activity. But in the case where that compound wasn't known, we would be able to say, okay, I know what this compound is, isolate it from the plant, and then do NMR or mass spec to identify that compound. Mm-hmm. So that cuts out, months or years of research time trying to find the compound you're interested in by just using machine learning. And so it's kind of a different variant of how you were saying you use Perplexity.
Yeah, kind of in reverse almost, in a sense.
Right.
Yeah, that's really interesting. Just out of personal curiosity, what was the compound that you found in basil?
It was actually gallic acid. Which is an incredibly common compound, and it has known cytotoxic effects. So it's, it was really convenient for my research to have, the compound that the model identified was one that was known to have that effect. So I could say, oh, look, my model worked. And then the next step would be applying the exact same model and research plan to a plant that's not as well known to see if we could identify a new bioactive compound that hasn't been seen before.
And for those, I'm guessing no one really knows this about gallic acid, but gallic acid is kind of a unique compound because you can use it to analyze other compounds, right? Mm-hmm. I think we use it in some of our methods too, maybe for EGCG kind of stuff, or yeah.
Yeah, I think so. And it's oftentimes used as a standard in antioxidant assays. So you'll say your compound or your plant has the equivalent of this level of gallic acid in an antioxidant assay.
So gallic acid is present in the basil plant itself, but
Mm.
gallic acid is also used as a material when identifying botanicals with these instruments. Do you know where this gallic acid is coming from when it's being used like for the purpose of identification? Like, is it being extracted from plants or is it being synthesized or?
I would say it's more likely synthesized based on the price of the standard.
Mm.
And it's not an incredibly complex compound, so it's probably pretty easy to synthesize.
Okay.
But it is found in most plants as well.
Okay. And maybe I misunderstood this, but did you say that gallic acid is toxic in any way, or did I mishear that?
It was in a higher concentration in the plants that were more cytotoxic, so ones that killed cells more.
Cytotoxic.
I wouldn't say
okay.
it is toxic, but at a high enough level, it had an impact on cells.
Gotcha. And cytotoxicity, can you give like a workable definition so I can understand what this is?
Right. So cytotoxicity is how well a extract kills cells, essentially. So if something is cytotoxic, it is killing cells. And that's in cell culture, so not necessarily in our body. It's going to react the same way. But when we have isolated cell lines that we're growing in petri dishes using an incubator, and you treat it with a plant compound, then we can measure if those cells die or grow, and that tells us if a compound is toxic or not.
Okay.
And that's what we're going to be doing with our new cell culture lab that we're starting up. And so hopefully we'll have a lot more data about these cells and be able to do some of this bioactivity research through Nootropics Depot and have more information about our compounds and our products that we sell. And I think we'll be able to start implementing some of these machine learning and AI tools in the use of this actual cell data, which will be really exciting. And I don't think any other supplement brand is doing anything close to that. So
No.
I'm really excited to be able to like take my thesis research and these AI tools and really apply it to making the best supplements we can possibly make.
And I think we can discover a lot of new things through machine learning. We recently had a call with a company that's developing a colostrum product, and they're also using machine learning to see which of the fractions, because colostrum is a bunch of different types of proteins, and you can focus in on specific ones, the glycoproteins usually, and different molecular weight ones have different effects on muscle growth, on immune function, and through a machine learning program that they used, they were also able to find different ones and focus on those and do the extraction on those and then do the clinical trial on those, instead of having to do clinical trials on potentially thousands of different glycoproteins, many of which will not meet the results that you would want to see. So being able to parse things out a little bit is really nice. Of course, the one downside is you might miss some things through that, but the upside is that you can actually do these things in a reasonable amount of time, and it doesn't take years and years and years, because there's still some plants that we don't really know exactly what the bioactives are in there, and then it makes it really hard to standardize for it. Or in the case of mushrooms, like we're testing for beta-glucans as a quality marker, but that's actually a really bad marker to be looking at, because every single mushroom has beta-glucans, but yeasts also have beta-glucans. There are a lot of other things that have beta-glucans, so they're not super unique. So being able to get more specific in the bioactives and what we're looking for is really nice. Because of course, it would be hard if we're trying to identify holy basil by its gallic acid content, because pretty much every plant on the planet has gallic acid in it.
That's actually a really good comparison of why beta-glucans aren't the best thing to use for identity of mushrooms, because you can't tell any difference between mushrooms based on that.
Yeah.
And to connect it back to what you had told us about gallic acid and basil, you discovered that this was a peak. Is that what you said? Or that this was present in higher amounts, correct?
Yeah, it's present in higher amounts in the ones that had more cytotoxic effects.
Okay. And so this is important because it helps you identify the the basil that has more cytotoxic effects specifically.
Right. So we could use it either as, say we had found a novel compound that hadn't been known before, we then would have found a new bioactive that maybe we could use that single bioactive compound in pharmaceuticals or as a supplement. But we can also use it, and this is interesting, in developing new cultivars. We could say, okay, I'm interested in this compound because I know it has this effect. I'm going to breed my plants to have more of this compound. And you could also do the reverse. Say you want lower gallic acid effects, so it's maybe having less of a negative effect on cells. You could say, I'm going to breed plants that have less of this compound. So it can guide breeding programs, product development, pharmaceutical development, and having these tools to really quickly identify the compounds can just speed up research altogether.
Okay, this makes sense because basically the gallic acid is like the indirect manner with which you can have more control over the plant that you grow or just know more about the plant itself. So this compound in and of itself may be not so interesting because it's in many different plants, but by being able to identify this compound quickly, you basically get the identity information that you need faster, efficiently, and then you can actually work with that information.
Exactly, yeah.
Okay. Yeah.
And ideally, we would also have been able to, when I was trying to classify the different species for authentication, if we could have found one compound that was like, all of the sweet basils have a really high amount of this compound, then we would have been able to use it for really quick identification schemes. So we could say, now I have a new basil, instead of testing all of the compounds, I'm just going to say, does it have a high level of compound X or not? And that's kind of what a lot of people are trying to do, and that would be the ideal situation. But there's just so many compounds in plants, it makes it pretty hard.
Yeah, really hard.
But perhaps over time, with more people gaining access to these, this machine learning or AI or just these tools, perhaps over time and as these things improve and become faster and just gain more knowledge themselves, maybe that will start to catch up. Like, the more people that use the tools, the more information that's put into them. It, of course, depends on whether people are sharing this information or not, and that's something that I'm not aware of because I don't use these tools. But perhaps then it aggregates a certain amount of knowledge to then, maybe predict more, or just it has a bigger database.
Yeah. The database is important, and I do think, academic researchers are sharing their data. So, like, all the data from my research is publicly available. You could download the data files if you wanted to. But then there are also companies like, there's a company, Purity IQ, that we work pretty closely with. We're doing some research papers with them now that have their own in-house databases that are really, really, really extensive. And the bigger your database, the more accurate it is. And it is, just like you said, over time, it's going to get better. And I think we're just right at the peak of it. And it's really exciting to be working with a company that's embracing it. And we're like right at the peak where things are about to start happening, I think. And I think we all kind of feel that.
Yeah, it definitely feels like it. And like you said, machine learning. The machine is learning. So it will get better and better over time. And I think one interesting thing to maybe touch on too is we're oftentimes struggling with what's the best extraction methodology, because that really depends on what compounds you want to get out. So for example, in lion's mane, in the fruiting body, we want to be able to get the hericenones out, but the hericenones seem to really not like any water being present. So once we can identify the hericinone is important, it doesn't like water, we're not going to do any type of water extraction on it. It's going to be an anhydrous solvent of some kind that is better at pulling those compounds out. And I think through machine learning, we can also maybe figure out how to do more and more advanced extractions with multiple different solvents, with multiple different techniques. And this is something we're actually using Perplexity for too now. Like I mentioned with Tangadali, this is one thing we're looking at. How can we best take a fresh Tangadali root and turn it into an extract that retains everything that's in that fresh root. Because from that root being pulled out of the ground, you can basically say that it's instantly starting to degrade. Once it's out of the ground, things are still happening in that plant, and those things probably are not good. So how quickly can you then get it into some sort of extraction facility? Maybe we don't even have to dry it. What challenges does that bring with it? What challenges does that bring with extraction? So I think for me that's a really exciting thing to look at with all of this machine learning, is how can we have machine learning extracts be really advanced extracts, because right now extraction is super rudimentary for the most part. You basically take your material, you dry it. Depending on where this is being done in the world, that material might have just been dried out in the sun, and it just in a big pile, and it gets turned over every once in a while and it just dries. But that means there's also a lot of UV-based degradation that's potentially happening, or UV-based creation of new compounds. It could also be a positive thing. And then with all of the extraction, you basically take that dried plant material and you dump it in a bottle of vodka, basically. Which would be a water ethanol extract, because vodka is about 40% ethanol, about 60% water. That actually seems to be a ratio that's being used pretty often in botanical extraction as well. I'll see that like in the study, 40% ethanol, 60% H2O. That sounds like you just used a bottle of vodka from a store. Which some people honestly might even be doing.
If it works, it works, right?
Yeah. And I think ethanol is just such a simple solvent to create. I think even the ancient Egyptians were distilling and creating ethyl alcohol that they then used to extract. I think this is actually an important concept in alchemy. I think it's kind of fun to read about sometimes. I don't really know if the concepts are so scientific, but it seems like in alchemy ethanol is really important, so they always use ethanol as the extraction solvent, but ethanol might not always be the best extraction solvent, or it might work better if you add some energy in the form of sonication, or if you dry it in a different way, or if you dry the resulting extract in a different way, or if you dry it at a lower temperature. Like we can maybe use a rotary evaporator, which now you can remove solvents from your extract at lower temperatures, and that might be positive. So I think being able to steer that with machine learning, such a complex process, and make it more refined would be really excellent.
Especially on the formulation side and like the creation side, but then if you look at it sort of on the back end or from the other perspective, you could also potentially use machines to measure the shelf life of a product, the efficacy of a product at a certain point of time, and you could also even compare maybe products that are extracted with different solvents and how those age over time. So it could also be something that you use once the product is actually completed as a way of just measuring it over time. And once you put in those time-sensitive properties, then you speed up that process by being able to predict degradation perhaps years down the road, which could probably be useful for a lot of different supplement companies.
Yeah, that's actually an idea I hadn't thought about. That maybe we should start trying.
And actually,
and I think
gallic acid could play a role there, because if a lot of plants contain gallic acid is a good antioxidant, so it would have some sort of antioxidant protective effect on your extract. But if your extraction technique doesn't include gallic acid, it leaves it out of the final extract, then maybe you have a more potent extract, but it's not as stable, and then, yeah, who cares?
And that's definitely something, even just this conversation right now, we're bringing up so many different points that it would be impossible for us to find all of the research about everything for every botanical. But things like Perplexity can say, oh, well, this hasn't been looked at in this plant, but here's a similar plant with similar compound class, and this is what has been done before. And so it can't do all of the research for us, so it can't, replace scientists, but it can really help speed along the research process and kind of help push new innovation forward without, the months of research that it would take otherwise.
Absolutely. For me, that's actually a good use of perplexity with this example that I pulled up earlier with that Canela de Velho plant. It's a South American plant. It's coming from Brazil. We have come to the realization that anything coming from South America, and especially Brazil, is hard to get a hold of. It's hard to be sure exactly what it is. It's easier to work with Chinese manufacturers, but a lot of Chinese manufacturers don't do anything with traditional herbs from Brazil. They usually deal with traditional Chinese medicine herbs. But through perplexity, I just put in what other plants also contain the emerins, and it pulled up a plant that is specifically used in traditional Chinese medicine for its analgesic and mood boosting effects. And it has a high amount of alpha and beta emerin, which this Canela de Velho plant has as well, and they seem to have overlapping uses on opposite sides of the world, but with identical bioactives in two completely different plants, one being endemic to Brazil, one being endemic to China. That's amazing, but it would have been really hard to make that connection without perplexity.
Definitely.
Wow. Okay, this is a really exciting moment to end the podcast on, because, like you said, we have come up with lots of new ideas. We've talked about lots of different varieties of plants, different ways to use machine learning and AI, and the goal is basically to improve and expand and deepen our understanding of botanicals and the quality of the supplements that Nootropics Depot is making. And it's so cool to have you on the team, Dr. Ellie Abraham, because you have so much passion, and the way that you explain these concepts is really clear. It's so clear that I'm able to understand the concept itself, even without having worked with the technology in my own hands. So thank you for that. And it's also really exciting to just hear all of the new projects that are going on at Nootropics Depot, a lot of which I am not aware of. So I hear it for the first time on the podcast. So for all of those of you who are watching, this is also new information, things you can look forward to. So stay a part of our ecosystem, subscribe to our YouTube channel, and subscribe to our subreddit r/Nootropics Depot. There's lots of conversations being had about our nootropic supplements, but also about technology and research and ideas and plants, so there's never a dull moment, and we love to hear from you. We're so grateful that you're watching the podcast and sharing it with your friends, and we're going to see you next month with a brand new topic and some more exciting research to share. So until then, we'll say goodbye for now, and thanks again, Ellie, for joining us on this podcast.
Yeah, thanks for having me. It was a really fun discussion.
Absolutely.
Awesome.
As always. And for everyone watching, this is a pretty good insight into how a lot of our meetings go at work. We really just do this for hours and hours and hours and talk to each other. A lot of our meetings will go upwards of 6 hours because we have a lot to talk about. And this is what a lot of those conversations feel like. And like Ellie was just mentioning, oh, I hadn't thought about that. I'm going to go look into that now. And this is how our calls always go. And then we're doing live research and we're plugging it into Perplexity, and it's all coming together, and it's a lot of fun.
And we definitely appreciate ideas from our customers and people listening to the podcast. And this spurred an idea, put it in Reddit, because then, we can't think of everything. So we appreciate when people help us think of new ideas or things that we never looked at before. Especially considering or thinking about the machine learning and AI. If you have a new use for it that might be useful for us, let us know. That'd be awesome.
Yeah.
Yes.
And in that sense, our Reddit is kind of an HI, a human intelligence, because we get a lot of new information from you, and it allows us to go on new research directions, and that's really beneficial. For example, cyanidin 3-glucoside was never on our radar, and it popped up on our radar because of Reddit, because people on Reddit told us to look into it. And now we can take that Reddit HI and match it up with the new AI. But
awesome.
I'm going off topic again. This mushroom magic baller is working well.
Totally. It's been
you
a
already
great
wrapped it up. So.
It's been a great conversational lubricant. So we'll leave it at that. Thank you again for listening, and we'll see you next month on the In Search of Insight podcast. And bye-bye for now.
See ya.
See ya.